Good afternoon, one and all. I am Shweta, Assistant Professor, Department of Microbiology, Jamal Muhammad College, Trichy. I am going to discuss about biofertilizer. What is biofertilizer and their applications? Generally, agricultural land gets improvised after long-term cultivation if not supplemented properly with inputs. To supplement the soil nutrient content under conventional farming system, we need to apply high dose of agrochemicals which in turn pollute the ecosystem. Therefore, in order to make agriculture sustainable, it is necessary to implement a balanced and responsible use of organic agriculture. The principles of organic farming also outline the similar concept where the soil health and biodiversity is built up to sustain the plant growth in longer term. Biofertilizer, it enriched the soil fertility and also it reduced the chemical fertilizer. What is biofertilizer? Biofertilizer is a ready to use live formulation of such as beneficial microorganisms which on application to seed, root or soil mobilize the availability of nutrients by their biological activity. They help build up soil microflora and thereby the soil health. As we know, organic farming excludes the use of any chemical. Use of biofertilizer is recommended for improving the soil fertility in organic farming. The classification of biofertilizer for nitrogen, for phosphorus, for enriched compost. And then what is the types of biofertilizer? Nitrogen fixing biofertilizer. The nitrogen fixing bacteria work under two conditions. One is symbiotically and another one is free living bacteria, that is non-symbiotic. The symbiotic bacteria make an association with crop plants through forming nodules in the roots. The free living bacteria do not form any association but live freely and fix atmospheric nitrogen. And then rhizobium. Rhizobium lives in the root hairs of the legumes by forming nodules. The name rhizobium was established by Frank in 1889. This genus has 7 distinct species based on cross-inoculation group concept. More than 20 cross-inoculations groups have been established so far. A new classification has been established for rhizobium, that is slow growing rhizobia and fast growing rhizobia. Still this classification is because the bacteria of one group may infect another group. This is called the principle of cross inoculation. And then method of application of rhizobium inoculants. The seed treatment has been found to be the suitable method of rhizobium inoculation. Some adhesive is used to make proper contact between seed and inoculants bacteria. About 900 gram of soil-based culture is sufficient to inoculate the seeds for 1 hectare area in case of legumes. A 10% agar solution is used as sticker for rhizobium cells to seed. First, the solution is spread over the seed and mixed to build a thin coat over the seed. After the proper coating of slurry over the seed, the inoculant is sprinkled over the seed and the content is again mixed thoroughly. Then content is dried in the shade by spreading thinly on a polythene sheet at least for overnight. This is a free living or non-symbiotic bacteria does not form nodules but make association by living in the rhizosphere. Azospirillum species establish association with many plants, particularly plants such as maize, sorghum, sugarcane, etc. It is the most common organism can form associative symbiosis on a large variety of plants. Azospirillum is recognized as a dominant soil microbe. And then blue-green algae. Blue-green algae is an important class of biofertilizer. The blue-green algae are small organisms and can be seen under the microscope as a single cell or large accumulation of cells or string of cells. They have a similar external appearance to that algae and azolla growing in a pond. The requirement for light, nutrients and carbon dioxide are also similar. And then Acetobacter. Acetobacter is a heterotrophic free-living nitrogen-fixing bacteria present in alkaline and neutral soils. Bacter chronococcum is the most commonly occurring species in arable soils of India. Apart from its ability to fix atmospheric nitrogen in soils, it can also synthesize growing promoting substances, auxin and gibberellin, and also some extend the vitamins. Many strains of Acetobacter also exhibit fungicidal properties against certain species of fungus. Response of Acetobacter has seen in rice, maize, cotton, sugarcane, and pear millet, vegetable, and some plant crops. Field experiment carried out on Acetobacter. And feature of Acetobacter contributes to the moderate benefits. Acetobacter is heaviest breathing organism and requires a large amount of organic carbon for its growth. It's poor competitor for nutrient in soil. It can benefit crops by nitrogen fixation, release of growth promoting substance, and fungicidic substances. Acetobacter is less effective in soil with poor organic matter content. It improves seed germination and plant growth. Acetobacter diacetrophicus is a newly discovered nitrogen fixing bacteria associated with sugarcane crop. This bacterium belongs to the alpha group of Proteobacteria. It was isolated from leaf, root, bud, and stem samples of sugarcane. Acetobacter is located in apoplastic fluid of sugarcane stem and to some extent in xylem vessels. It is an acid and high salt tolerant and sucrose loving bacteria which can fix up to 200 kg nitrogen per hectare. And then next, VAM, vesicular arbuscular mycorrhiza, was taken from Greek language meaning fungus root. The term was coined by Frank in 1885. As indicated above, the mycorrhiza is a mutualistic association between fungal mycelia and plant roots. VAM is an endotrophic mycorrhiza formed by aseptated Phycomycetes fungi. VAM helps in nutrient transfer mainly of phosphorus, zinc and sulfur. They also mobilize different nutrients like copper, potassium, aluminium, manganese, iron, magnesium from the soil to the plant roots. They possess vesicle sac-like structure for storage of nutrients and arbuscular for funneling the to root system. And then mechanism of action, the VAM forms an association with plant roots. It penetrates in the root cortex and spread around the roots of the plant. As the name indicates, the possess sac-like structure called vesicles which store phosphorus as phospholipids. The other structure called arbuscular helps bringing the distant nutrients to the vesicles and root. And then method of biofertilizer inoculation, the seed inoculation. This is the most common practice of applying biofertilizer. In this method, the biofertilizer are mixed with 10% solution of jacquerie. The slurry is then poured over the seed, spread on a cemented floor and mixed properly in a way that a thin layer is formed around the seed. The treated seed should be dried in the shade overnight and then they should be used. Generally, 750 gram of biofertilizer is required to treat the legume seed for 1 hectare area. Soil applications. Soil application, this method is mostly used for fruit crops, sugar cane and other crops where localized applications are needed. At the time of planting of fruits trees, 20 gram of biofertilizer mixed with the compost is to be added in the ring of one sponge. Sometimes the biofertilizer are also broadcasted in the soil, but we may require 4 to 10 times more biofertilizer. Before broadcasting, the inoculation should be incubated with the desired amount of well decomposed granulated FIM for 24 hours. Self-inoculation of tube-based inoculation. And then what are the disadvantages? Disadvantages, as such, there is no harmful impact of biofertilizer if it is used properly. Some methods and condition. Main disadvantages is difficult to store. Thank you.